Hey there, science fans. Have you ever wondered how your cells know what to do? I mean, they're tiny, but they build your tissues, fight off infections, even think your thoughts. Well, it all comes down to which genes are turned on and off. That's gene expression. It's like a secret language your cells use, and today, we're going to learn how to decode it. We'll see how scientists use gene expression analysis to diagnose diseases, personalize medicine, and even understand how life itself works. Get ready to explore the amazing world inside your cells. Alright, science crew, let's start with the basics. Every single cell in your body from your nose to your toes contains the same DNA blueprint. That's incredible. But if they all have the same instructions, how come a heart cell doesn't try to pump blood in your brain? The answer is gene expression. Think of your DNA like a giant cookbook. It has recipes for everything your body could ever need. But you wouldn't cook every recipe at once, would you? That's where gene expression comes in. It's like choosing which recipes to make and how much of each one. Each cell only turns on or expresses the genes it needs for its specific job. So, a heart cell reads the heart muscle recipe, while a skin cell reads the keratin production recipe. Pretty neat, right? Now you might be wondering, why do we even care about analyzing gene expression? Well, it's like listening in on your cell's conversations. By studying which genes are turned on or off, scientists can learn a ton about what's happening inside your body, both in health and disease. For example, cancer cells have messed up gene expression patterns, like a recipe gone wrong. By analyzing these patterns, doctors can diagnose different types of cancer and even predict how aggressive they might be. Gene expression analysis is also revolutionizing how we treat diseases. It helps us understand how patients respond to medications and even personalize treatments for better outcomes. It's like having a personalized health report straight from your cells. So how do scientists actually measure gene expression? Well, they've got a few tricks up their lab coats. One common method is called Rt-PCR. It's like a high-tech copy machine that specifically amplifies and measures the RNA transcripts of a gene. The more RNA copies, the more active that gene is. Then there are microarrays, which are like tiny chips that can measure the expression of thousands of genes simultaneously. It's like comparing a whole library of recipes at once. And for the ultimate deep dive into the cellular cookbook, there's RNA sequencing, or RNA-Seq. This technique sequences all the RNA in a sample, giving us a complete picture of gene activity. It's like reading every single recipe in the book. Now let's walk through the steps of a typical gene expression analysis experiment. First, we need to collect our biological samples, like tissue, cells, or blood. Then, we extract the RNA, which is like carefully removing the recipe pages from our cookbook. But RNA is delicate, so we convert it to a more stable molecule called cDNA. Next, depending on the method we're using, we might add fluorescent labels or barcodes to our cDNA. For microarrays, we let the labeled cDNA bind to the chip, and the brighter the spot, the higher the gene expression. For RNA-Seq, we sequence the cDNA and use powerful computers to analyze the data. Finally, we visualize the results using graphs and charts to see which genes are turned up or down. Okay, so we've got our gene expression data. Now what? Well, it's time to put on our detective hats and interpret the results. We look for genes that are upregulated, meaning they're more active, or downregulated, meaning they're less active. Scientists use fancy graphs called volcano plots and heat maps to visualize these changes. Volcano plots show us which genes have the most significant changes in expression, while heat maps give us a big picture view of gene expression patterns across multiple samples. By analyzing these patterns, we can start to understand the biological processes that are affected by a disease or treatment. Gene expression analysis is like having a superpower that's transforming many fields of science and medicine. In cancer research, it's helping doctors develop more targeted and effective treatments. In pharmacogenomics, it's helping predict how patients will respond to different drugs, paving the way for personalized medicine. And in infectious disease research, it's helping us understand how viruses like COVID-19 hijack our cells and how our bodies fight back. Gene expression analysis is even being used to study stem cells, those amazing cells that have the potential to become any cell type in the body. It's a truly revolutionary tool that's helping us unlock the secrets of life itself. Now, while gene expression analysis is an incredibly powerful tool, it's not without its limitations. Remember how delicate RNA is? Well, that means we need to be super careful when handling samples to avoid degradation. And even with the most advanced techniques, there's always a chance of experimental error or biological variation. Plus, analyzing huge datasets from RNA sequencing requires some serious computing power and bioinformatics expertise. But hey, that's science for you. Always pushing the boundaries and finding new ways to overcome challenges. Gene expression analysis is like listening to the cellular symphony of life. It allows us to decode what cells are doing, why they're doing it, and how we can intervene when something goes wrong. The future of diagnostics, therapies, and precision medicine all starts here with the language of genes. Like and subscribe for more deep science made simple. Comment below, which gene expression method are you most curious?
Imagine the vastness of the Amazon rainforest, teeming with life in countless forms. Each organism, from the towering kapok tree to the tiniest ant, carries within it a set of instructions that determines its traits and behaviors. This instruction manual, passed down through generations, is the genome. Now picture intrepid explorers venturing into this dense jungle, meticulously charting the landscape, identifying landmarks, and mapping the intricate network of rivers and tributaries. This, in essence, is the ambitious endeavor of genome mapping. Just as a map helps us navigate a complex terrain, a genome map guides scientists through the labyrinthine world of DNA. This blueprint of life holds the key to understanding the very essence of who we are, from our physical characteristics to our susceptibility to diseases. By deciphering this code, we unlock a treasure trove of knowledge that has the potential to revolutionize medicine, agriculture, and our understanding of life itself. The genome, an organism's complete set of DNA, is akin to a vast library containing thousands of books, each representing a different gene. These genes, like individual chapters within a book, hold the instructions for building and maintaining a living organism. Genome mapping, in its simplest form, is the process of creating a detailed map of this vast library. It involves identifying the location of genes and other genetic landmarks within the genome, much like pinpointing the locations of cities and towns on a geographical map. This map serves as a valuable tool for researchers, allowing them to navigate the genome, identify specific genes of interest, and understand how they interact with one another. Imagine trying to navigate a sprawling city without a map. It would be a daunting and inefficient task. Similarly, attempting to understand the complexities of human health and disease without a genome map would be like searching for a needle in a haystack. Genome mapping provides the crucial framework for understanding the genetic basis of diseases. By pinpointing the location of genes associated with specific conditions, researchers can develop targeted therapies and diagnostic tools. It also sheds light on the intricate mechanisms of inheritance, helping us understand how traits are passed down through generations. Genome mapping encompasses two main approaches: genetic mapping and physical mapping. These two techniques are like two sides of the same coin, each providing a different perspective on the genome. Genetic mapping, analogous to creating a roadmap based on travel time between cities, determines the relative order of genes on a chromosome by analyzing how frequently they are inherited together. The closer two genes are located on a chromosome, the more likely they are to be inherited as a unit. Physical mapping, on the other hand, resembles measuring the actual distances between cities on a map. It involves analyzing the DNA sequence itself to determine the precise location of genes and other genetic markers. Both genetic and physical mapping contribute valuable information to our understanding of the genome, working in tandem to provide a comprehensive view